All right, Peter Innes is a PhD candidate in the Ecology and Evolutionary Biology Department at CU Boulder in the Kane Lab. He studies evolutionary genomics of crop wild relatives, including sunflowers and blue flax. Before graduate school, Peter worked at the Rocky Mountain Biological Laboratory outside of Crested Butte, Colorado, as a summer research technician and winter caretaker. To learn more about his work, you can find him on Twitter at Peter Innes or visit peterinnes.org. And for spellings of those, you can look onto the website for that. All right. Thank you, Peter.
Hey, hi everyone. First, I just want to thank Maggie and the organizing committee for the invitation. I'm really excited to talk to you today about some of my dissertation research on the sunflowers that live in Great Sand Dunes. So I think this is a perfect venue for my work. I don't think I need to convince you that the dunes are a really special place, and I feel really lucky to get to I just feel really lucky to get to work here. It's a really unique ecosystem, and my understanding is that it's really significant to multiple indigenous peoples and nations in the region. So the Ute people have a name for the dunes that translates to sand that moves. And yeah, I really love that and just wanted to share it with you. So today I'm gonna just give some background on the ecosystem in the dunes and what we know about the sunflowers there. And then in the second part of my talk, I will discuss some of my own research on the genetic basis of their adaptation to the sand dunes and the role of gene expression. So it's a bit of a shift in topics, I guess. We're going to be talking about evolution and genetics now. So the sand dunes are pretty inhospitable, relatively speaking, to life, and they're very windy. There's a lot of exposure. Surface temperatures can get up to 150 degrees. There's really low nutrient levels, especially nitrogen, which makes life for plants especially hard. But a few species do make their home in the dunes. So here's a few of the plants that you'll find in the dune field. There's a legume, a scurf pea, a couple of species of grass, including the Indian rice grass or sand rice grass, this rush skeleton plant, and of course, the star of the show today, these prairie sunflowers, which we refer to as the dune ecotype. But so I also want to mention some of the animals in the dunes. There's only one endemic mammal in the dune field. It's this really cute Ord's kangaroo rat. There's multiple endemic invertebrates like the Great Sand Dunes tiger beetle. There's also these giant sandworms. They make fieldwork quite treacherous. Here's a photo of me in the dunes. Anyways, okay. So back to the sunflowers. They weren't really characterized by science until around 10 years ago. And initially, we didn't know, or I should say my colleagues didn't really know what they were because they looked pretty different than the prairie sunflowers in the surrounding San Luis Valley. And so some initial hypotheses regarding where they came from included, one, maybe it was just Helianthus annuus, the common sunflower, which it's closely related to. Or perhaps it was a hybrid species resulting from, cross between annuus and Pediolaris, because this had actually happened in the past in other parts of the southwestern US, giving rise to Helianthus anomalus, which is also dune adapted. And also it was thought maybe it was They were in the dunes resulting from long-range colonization from New Mexico, where this subspecies of Pediolaris canescens inhabits sand dunes. So to answer this question, a group of scientists from University of British Columbia, led by Dr. Rose Andrew, compared genetic markers from Helianthus annuus, from the great sand dune sunflowers, and from other Pediolaris subspecies. And this is what they found. So this, I'm showing you results from what's called a principal components analysis. And basically, it's summarizing genetic variation across the genome of a bunch of individual plants. So each dot or label here is a plant, and some of them are labeled according to the Pediolaris subspecies or according to, AN, that's annuus. And so the genetic variation summarized into these two synthetic axes that are independent. And if the dots are closer to each other, that means they're more genetically similar. So as you can see, the great sand dune sunflowers are clustering on their own. They're more similar to each other than they are to annuus or any of the other subspecies. And so that cluster there in the bottom left has both the dune plants and we call them the non-dune plants that grow around the periphery of the dunes. And when they, made a evolutionary tree from the same samples, you see the same pattern where they formed their own clade, and their closest relative is Helianthus pediolaris subspecies phalanx in black there. And so that's what they're labeled today as subspecies phalanx. And yeah, I guess just a unique ecotype. We call it the dune ecotype of subspecies phalanx. So yeah, in the process of adapting to these really distinct habitats, we're in the dunes, it's really extreme abiotic pressures, low nitrogen, compared to off the dunes where there's more competition, perhaps. The two ecotypes have evolved really different strategies overall to growth and reproduction. And this is perhaps best illustrated by a difference in seed size, where the dune plants have seeds that are over twice as large as the non-dune plants. And this is maintained even in a greenhouse common garden setting. The dune plants also tend to be just more robust plants. They're larger, thicker stems, larger leaves. And seed size in particular has been shown to provide a really strong adaptive benefit in the dunes. So Dr. Kate Ostovic did her PhD on this system as well back in 2016 and did a reciprocal transplant experiment growing both ecotypes in either habitat and basically found that the large dune seeds improve emergence of seedlings in the dunes. Whereas off the dunes, it's more beneficial to have or to produce a lot more seeds, even if they are smaller. And an important point I want to make is that this divergence is occurring despite the fact that the two populations are in very close geographic proximity, and they're still exchanging genes. There's still some cross-pollination going on. And so this really implicates natural selection as the driving force in their divergence, because gene flow acts to kind of like make things the same. It homogenizes any differences that arise. But the fact that we do see them, maintaining divergence suggests that natural selection is quite strong. And this is illustrated at the genetic level as well. So this is what we call this figure here is it's called a Manhattan plot because it looks like the Manhattan skyline kind of. And what you're seeing is DNA sequence divergence across the genome. So the x-axis is just genomic position, all the 17 chromosomes of the sunflower, and the y-axis is a measure of sequence divergence or differentiation. And so across most of the excuse me, across most of the genome, the divergence is relatively low, except for a few regions that you see those skyscraper peaks. And so, the gene flow is homogenizing things for the most part, except for the regions where, we think that are most important for adaptation in the dunes. And for what it's worth, those colored blocks, at least the blue squares above the peaks, correspond to regions that are known to be associated with the seed size trait. So there's some correspondence there. And so all this begs the question, well, how is this divergence, how is this differentiation being maintained? And I mentioned natural selection was really important, but Kate Ostovic, who I mentioned earlier, did some more work into quantifying multiple potential barriers to reproduction between the populations. And this is what she found. Here's a summary. So she quantified a bunch of different barriers, like I said, both things like flowering time, things like selection against non-dune plants moving into the dunes, selection against hybrids, and so on. And so the y-axis is a measure of barrier strength, and you can see different barriers are, some are stronger, some are weaker, but some of the strongest barriers have to do with selection against non-dune plants coming into the dunes. Whereas things like flowering time, there's not much of a difference. There does appear to be some difference in pollinator assemblage, which is interesting, as well as post-pollination assortative mating. So that has to do with precedence of pollen, or for pollen of the same type. Okay, so that's all I have for this first part. I guess in summary, the Great Sand Dune sunflowers were really, I guess, surprising in where they came from or what they were, and that they represented an independent origin of the sand dune adaptation, which had been seen in other sunflowers in North America, but they evolved in place here in the San Luis Valley. And natural selection is really strong. That's the driving force here in maintaining the differences between the dune and non-dune plants. And lastly, isolation between the two populations is not complete, so you can still cross them. They're pretty interfertile, but there are some reproductive barriers that have begun to evolve. And so while they are not distinct species right now, we think they are like moving in that direction. So check in another 10,000 years and I'll let you know. Okay, so I'm going to move on to part 2 now, which is some of my own research. And so I'm an evolutionary geneticist, and so I'm really interested in the genetic basis of novel phenotypes like the dune plants and the basis of adaptation. And that kind of question often leads us to study gene expression. So gene expression is this, fundamental link between genotype and phenotype and the environment. And it's comprised of a lot of complicated mechanisms here, right? But you can basically think of it as how genes are regulated in the cell, whether they are turned off or on, when they're expressed, where they are expressed, and to what extent. And so that's why, even though you have all your cells in your body have the same DNA sequence, they form a vast array of different tissues, right, leading to a complex organism. All of that is thanks to gene expression. And so I'm going to simplify things a bit and just talk about a couple of gene expression mechanisms today, starting with transcription. So this refers to basically the copying of DNA into RNA, which relates to how much of a gene is expressed in the cell. And so you can think, an analogy I like to use for this is a giant mixing board. So think about if you're like a sound engineer and you have a volume dial for like 30,000 genes, and you can turn some up, some down. And that's a bit like, what gene expression looks like in the cell. So some genes will be expressed more or less in certain conditions or in certain environments. Yeah, and this can ultimately change how physical traits, manifest at the level of the individual organism. But today I'm going to be also talking about this mechanism of RNA splicing, which we know a lot about the molecular mechanism here, but we really don't know much about how splicing contributes to adaptation and divergence. So when you hear splicing, you might think of some sort of like gene editing technology, but this is something totally unrelated. This is a process that naturally occurs in basically every eukaryotic organism. And it's because our genes are coded in pieces in the genome, and those pieces are called exons. In this figure, they're the like larger colored blocks, and they're interspersed by introns, which are the thin lines. And before the RNA molecule is translated into protein, which is kind of the molecular machines of the cell that carry out all the necessary functions for life, that RNA molecule has to be processed such that the introns are removed and the exons are joined back together to produce the what we call the mature messenger RNA molecule. And this is performed by this protein and RNA complex called the spliceosome. And so RNA splicing occurs for every gene that has multiple exons, okay? But in some cases, alternative splicing occurs when those exons and introns are included or excluded in different combinations, giving rise to multiple different versions kind of the same gene. And we call those isoforms. So an analogy I like to use here is like, it's like you're a film editor and you're splicing together different scenes to produce, two different versions of a film. And, maybe one is a lot longer, one's more shorter, and it's the same overall film, but they have like a different feel to it potentially. So it's like the director's cut versus the standard edition. And those different versions might be, thinking about the genetic level, the different isoforms might be more or less beneficial in a particular environment. Just like, if you go to the movie theater, maybe you want to see the shorter Lord of the Rings version because you don't want to sit in a movie theater for 4 hours. But at home, when you're comfortable on the couch, yeah, you're going to watch the extended edition because you're a huge fan. So yeah, that's alternative splicing. And an important point is that this process of splicing can be regulated by cues during development. And it's also really important in abiotic stress response for plants because, they can't move. So they'll change the splicing pattern of a gene, potentially depending on what they're experiencing from their surroundings. But also splicing can change if a certain genetic mutation arises, and that can influence which version of that gene is expressed and to what extent. And so that means this mechanism, this process, has the potential to contribute to adaptation. It can evolve itself. Okay, so this leads me to my main question in my work, which is, what role does this mechanism of alternative splicing have in adaptation and divergence, specifically for these sunflowers? And our experimental design here was pretty straightforward. First, we collected plants from on and off the dunes, shown here. And then we took them into the greenhouse and we grew seedlings. And this is an important part of the experiment because our goal here was to reduce variation in gene expression due to the environment. So they're experiencing a common relatively benign environment here in the greenhouse. Before we harvested them for our molecular methods, we measured some of the seedling traits. So hopefully you can see this, but the dune plants in yellow tended to be taller after 3 weeks. They had the same number of leaves, so developmental stage was about the same as the non-dune plants, but they also had greater mass and larger leaves. And so this is largely confirmatory. We already knew that the dune plants tended to be more robust. And you can imagine, you have a larger seed. It makes sense a larger seedling is going to result from that. So next, we harvested the tissue and extracted all of the RNA in those cells from that tissue. And then we sequenced that RNA. So we had 12 samples from each population. And this produces a ton of data. So you have it produces millions of RNA fragments, basically just strings of letters, A, T, G, and C, which you then align or match up to a reference genome, in this case, the common sunflower genome. And this lets us estimate how many RNA fragments match up to a particular gene. So that's a estimate of its overall expression level. And using what we call a splice-aware algorithm, we can also measure the alternative splicing pattern for each gene. And then, I did several analyses, which I'm going to tell you about next, basically just looking for differences between the dune and non-dune plants in transcription level, and also differences in alternative splicing and DNA sequence differences as well. That's the variant calling part. Okay, so first of all, we confirmed that the two ecotypes, the two populations are genetically distinct. This is to be expected. This is another principal component analysis plot. So points or individual plants, and if they cluster close together, that just means they're more similar in this data, which in this case was the genetic variation. And then we looked at variation in transcript levels, and we see the same sort of clustering. And likewise, this pattern holds when we looked at patterns of alternative splicing. And so, this was a pretty striking results. And basically, because natural selection is so strong in this system, we think that these mechanisms, the transcription and alternative splicing, could either be directly or indirectly under selection and could be contributing to their adaptation to the dune system. Next, we just considered or wanted to know more about the genes in particular that were really significant in their differences for expression pattern and splicing patterns. So DE, that's short for differential expression. Sorry, it's not written out in blue there. And then in red, that's the genes that were differentially spliced. So there are more genes differentially expressed in blue there, but still quite a few genes that were really divergent in their splicing pattern. And so next we wanted to know, well, what are these genes doing? What's their function? So first, looking at the genes that had different splicing patterns, this figure here is showing results from a functional analysis. We call it a gene ontology enrichment analysis. That's not important to remember, but each label there is a different functional annotation, and basically and they're arranged in descending significance, with the large circles being functional term that was really significantly enriched in the gene set, so the differentially spliced genes here. So of note is that we find some functions that are really prevalent in the differentially spliced genes that line up with some known adaptive traits. So this embryo development ending in seed dormancy, maybe that's related to the difference in seed size there. And we also see some functions related to nitrogen assimilation. So that figure there is just showing the differences in nitrogen in the dunes, so D versus intermediate and non-dune habitats. And then if we look at the genes that had different expression patterns, we see some similar terms. So nutrient reservoir activity, response to water deprivation, that kind of matches what we think the seedlings and the dunes might face, limited water and nutrients, as well as this response to abscisic acid. That's an interesting one because that's a key plant stress signaling hormone. And lastly, if we look at genes that are both differentially expressed and differentially spliced, we see this same functional term relating to embryo development show up. So that's an intriguing overlap in these mechanisms. Okay, that's, I think I'll say one more thing and then I'll end, but we also found one gene that showed one of the strongest differences in splicing between ecotypes, and it was related to lateral root development. A figure here just kind of illustrates that process. But on the right side, in the panel A, those are the two versions of this gene, the two isoforms. So the box in red is just highlighting the exon that is being either included or excluded. And in the dunes, in the yellow triangle there, it tends to be spliced out compared to the non-dunes. And so we think this gene in particular should be investigated in the future, or at least, root traits in general, I think would be a fruitful avenue to characterize for the dune plants, because no one's really looked at that yet. And you can imagine, especially for dune seedlings, having more stable root system or being able to access really limited nutrients could be really important for their survival. So with that, I'm going to skip this and just say thank you, and I'm happy to take questions if there's time.